Good morning, everyone, or good afternoon. We're delighted that you could join us. I'm Michelle Mannion from the PCD Foundation, and running our webinar today is Lynn Ernie, who many of you know.
We'll talk about the eligibility, the enrollment process, what to expect, and how can you learn more about it, and aim to have at least 20 minutes Q&A. So the next slide, please. And then, so we're going to talk about the prevalence and genetics of PCD. So PCD is a rare disease, but it is misdiagnosed and we think vastly underdiagnosed. This is in part due to the symptoms of PCD, which can be varied and involve things like sinus infections, respiratory infections, but it's also because of a lack of diagnosis. There's only recently been a consensus on the diagnostic methods, and many of these may not have been available except at very specialist centers. And so there's efforts to try and develop better diagnostic testing so we can get that more at scale out more into the community. The identifiable disease-causing variants are anticipated in 65 to 80% of PCD patients, which means a comprehensive genetic testing panel, along with improved screening questionnaire, may improve the diagnosis of PCD and ultimately treatment. So that's our aim, is to be trying to resolve some of these issues because of these problem statements. And with the potentially very large number of undiagnosed patients in the US, 46,000 plus, perhaps 75,000 plus undiagnosed in Europe. And we also have some evidence from the bronchiectasis community that we think that between 10 and 35% of patients in community clinics where bronchiectasis is looked after and in asthma clinics, there are these 10 to 35% of people have undiagnosed PCD that are being looked after in these clinics, but they're not being given specialist PCD care. So there's no gold standard for diagnosing PCD. It needs some clinical acumen to be thinking of it and to advance the genetic testing to get the most precise, clear underlying diagnosis of each case of PCD. And as we're saying, at least 65 to 80% of PCD patients should be able to get an identifiable disease-causing variant with our current understanding. Patrick, do you have anything you would like to add here?
No, nothing to add at this point. I promise I'll talk a little bit more later, but I'll definitely let you get through some of the background.
Cool.
Great talk.
So next slide. So here we've got some schematics to highlight the genetics of PCD. It's typically inherited with autosomal recessive pattern. That means inheriting the two changed genes from your parent. There's some exceptions. There's excellent cases. There's a change in the gene located in the X chromosome, meaning that females can be carriers, but then the disease shows up in males because they only have one X chromosome. But those are rare cases, but we recognize them. There are currently 50 genes known to be associated with PCD. Within each gene, there are multiple variants and mutations which can cause that gene product, the protein that comes from that gene, to be misfunctioning. You need the same gene on each allele. These are biallelic mutations to mean that that specific gene is not functioning. So enough from one side of the gene, one allele, can rescue the function as what we call a heterozygote. So it's biallelic mutations where there's full loss of function of a gene. And so, for example, within DNA1, there are 130 known pathogenic mutations. We call them pathogenic because it's clear that when those mutations on each side, on both sides of the gene, are these specific mutations, that leads to the function not working in the cells in the body as a product of those genes. And so it's multiple genes, mutations, but it's the single gene that loses function. And then of those, there are 50 of them that can then lead to disease-causing variants. And in terms of prevalence, DNAH5 is the most common, and I think that's very well established. 29% here from the data that we're quoting. DNAH11, 9%, DNAI1 up to 10%, and then CCDC3940 up to 9% for CCDC39 and up to 4% for CCDC40. I think the genetics is interesting. There's been some recent papers. Bill Hanna produced a paper where he went back to some genetic testing panels and looked at the genetics, and I think was able to show that potentially there's a higher prevalence than we're recognizing, and this is coming through as we do more and more genetic testing. So I'll move on to the next slide. We're going to highlight ReCode's investigational therapy, and to the next slide. So ReCode, we're working on developing an inhaled mRNA treatment for people with DNAI1 PCD. About 10% of people with PCD are missing this protein called DNAI1, which sits on the outer dynein arm and means that the cilia are not moving in PCD people with DNAI1 because that the movement can't happen with this outer dynein arm protein not being present. And so the mRNA treatment that is being developed needs to be specific for this missing protein. So it has the full instruction to produce DNAI1. And the schematic there shows that the mRNA is protected by a lipid particle. And so it's able to get into the body through a nebulizer. It comes in a mist that's breathed into the lung. And then is in the airway cells, it transfers into the airway cells. The lipid particles are able to bring it into the cell, protected. And then once it's inside the cell, the mRNA can be transcribed and the protein produced that then gets incorporated up into the ciliary axoneme, up into the structure of each cell. And we think we probably only need one mRNA per cell, one or two, to produce enough protein that then allows functional restoration of activity. And so the treatment will be a 3 times a week treatment we plan that would allow restoration of function by bringing in the instruction to produce the missing protein. And so this is where we are at the moment. And the plan, the therapeutic aim is to restore ciliary function in the lungs, getting at the root cause of PCD rather than just symptomatic control of the problem. So the next slide, we're in phase 1. The end of December, we were able to dose our first patient in the United States. The IND cleared. We had the conversation at the webinar to be able, an IRB approved presentation that was able to give the information about the study, and we had great response, and we are planning our second patient to be dosed next week. We are looking to complete 3 patients in that first cohort as soon as we can, and things are going well. It's an open label study. Everyone in the study receives the study drug, RCT-1100, as a single dose, and it's given as this nebulizer, and then the study visits are set up for safety. The first visit's an overnight visit, and then it's through day 3, day 8, 15, and 29. And we've arranged for remote visits so that the safety can be done closer to home. It's really looking at lung function and some blood tests, which are set out in the protocol. But the goal of the study is to evaluate the safety and tolerability of the single dose, to determine if there's a response in the body to the drug, its components, and to the mRNA in terms of an immune response. But once we have single dose safety, then we'll be able to advance and give multiple doses. We have preclinical toxicology now that once we have filed that to the IND, we'll be able to start a repeat dose study. And the aim is to then start to get airway samples or to look for mucociliary clearance as we see the functional effects of this therapy and assess what we call the benefit risk. What's it achieving and then what are the safety risks, so we understand the safety profile of this medication, potential medication. So the next slide. We can advance one more. So now we're moving on to the ThinkPCD. So really what we're doing with ThinkPCD.com is we want to have a broad genetic testing panel for all of PCD, such that as we help solve for this problem of really lack of genetic testing, we can then allow us to understand who has PCD, what's the exact genetic diagnosis for their particular PCD, and then really build up eligibility for clinical trials as we're able to bring through therapeutics, because the DNA one is kind of just a first start. The aim is to be able to replace out the mRNA and solve for the loss of function with mRNA across the different genes, the different loss of function genes, and really try to solve for as much of PCD as we can, frankly, in the shortest possible time frame. So ThinkPCD.com is a program to provide genetic testing to patients that qualify. It's at no cost. It's no direct cost to the participant. There are two ways to enroll. You can either come in as a healthcare provider and nominate one of your patients to come in and enroll, and we'll go through the process of how that happens, or you can come in directly as a patient. And I think that's a very exciting part of this platform, as it really aimed and designed to be able to have people come directly to it and go through the process. So that's what today's about, is really help you understand what this process is. So if we go to the next slide. So who's eligible? We're starting at the moment with individuals who are 18 and above, so adults. And there are three ways. Currently, you can have a PCD diagnosis and previous genetic testing. So if you're already aware of your genetic testing result, the platform allows that to be uploaded, reviewed, and then to participate in the program. You can have a PCD diagnosis, but then not have a genetic test, and that is also part of the participation and has a different pathway. You can also come in if you're strongly suspected of PCD, or you have a family history of PCD. And so this is where we're trying to bring through these people who may not know that they have PCD. They may be in a bronchiectasis clinic or an asthma clinic, but there's some suspicion that they have PCD. Either coming directly to the platform or through your healthcare provider, we can help assess those and get some more testing to deal with that 30% that I talked about earlier. So the next slide. So why would you participate? So you would receive no-cost genetic testing, which could confirm a diagnosis of PCD and the precise genetic driver of your PCD. The program gives you the opportunity to get genetic counseling. No matter which variant is identified, you would be connected with a genetic counselor to help you understand the result that you received. You can contribute to future PCD treatment options by participating in a clinical trial, because you have DNA1 facilitates that program. But the idea is, we call it, we have a waiting room, and the idea is that you would be contacted, you would consent to be contacted, and you could be contactable as your variant then has a treatment available, such that we can bring the treatment through by being able to really know exactly how many people are ready for clinical trials and how we can get those programs resourced and set up. You can receive alerts for future research opportunities relevant to your specific genetic variant. And lastly, there's access to expert condition-specific educational materials that would be available by being in the program and consenting to receive them and to go into the portal. So the next slide.
John, I might add just a point or two on this one, if that's okay with you. And
Sure.
I'll try to answer one of the questions in the chat at the time, which was about having either confirmed, well, there's two actually good ones. One is about having a confirmed diagnosis already, and the other related one is about other panels that exist. So I just wanted to highlight again that if you've already been tested, it's really easy through the program to just upload that result into the program, and that will make you, in this virtual waiting room and able to get notified about the RCT 1100 that John mentioned. Or if your variant is not DNAi1, then you can get alerted of future research opportunities. But on the panel question, if you have access to a comprehensive panel through the doctor you're already seeing or some other route, again, that's absolutely fine. And we'd encourage you to go take that up and then bring the result to the program. However, many panels are not very comprehensive actually, and don't include the full set of genes that have really strong scientific basis for inclusion. So we've got, I believe, as of the last approval of the protocol, 42 genes on this panel, including some that are particularly tricky to sequence that not all providers have. And that was really important for us in this program to make sure we were as comprehensive as possible. And the second is the breadth of people who can take part. So it's often very tricky to get access to genetic testing unless you have quite good insurance or have a healthcare provider that's really in the know of how to access them. So the inclusion criteria for this are broader, so that people who may have a suspected diagnosis, for example, can get access as well. So hope that helps and answers a couple of those questions in the chat.
Yeah, no, that's great. Thank you. Thanks, Patrick. So if we go to the next slide, this is what the process looks like for healthcare professionals who are seeing patients who may be eligible to participate. The healthcare provider will go onto the website and complete a questionnaire, which contains multiple questions about their patient's medical history and help to determine if they're eligible for genetic testing. If the patient is eligible, they'll receive a unique patient code, which links the patient to their healthcare provider and the information that the healthcare provider provided within the questionnaire. They'll give this code to the patient, who will enter it onto the website, and the content to participation, the consent to participation is completed, and then with that consent, they then receive the at-home kit. So it's a sequential process, but it allows the healthcare provider who has access to the medical records to go through the screening questionnaire, which is really just try to enrich for the clinical characteristics that we understand around PCD that will help us determine that this is a worthwhile route to go down. I'll go to the next. We'll go through the three flows, and then I think better to have Q&A. So this is the process for people with PCD and no genetic testing, and no, but they have suspected PCD. They could also have a family history of PCD to participate in this flow. So for a patient with confirmed PCD but no genetic testing, you enroll in the program, complete the online informed consent, complete a brief screening questionnaire, and then you'll receive the at-home genetic testing kit. For an individual with suspected PCD or a family history of PCD, the steps are basically the same, but you'll need to provide your HCP contact details before you receive the kit. And this is really just to help link up and make sure that we've got the most information to support your application and through your consent. And then the next slide. This is the process for people with PCD and a previous genetic test. So starting with on the left-hand side, firstly, enroll in the program, complete an online informed consent and a brief screening questionnaire. Would then allow you to upload your existing genetic test results. And then if the genetic testing is a clear certified lab and DNAi1 is present, then that would be a notification that you'd be referred directly to the clinical trial site that was available at the time. If it's not clear certified and perhaps it's ambiguous for DNAi1, then you'd receive an at-home testing kit. And then that would allow the potential through the genetic testing to then have any other mutation to be found or assessed, the link to the counseling and the process into the what we call the waiting room. So then the next slide gives you a sense of what you would expect once you receive your kit. The genetic testing kit would be sent to your home and would have detailed instructions on using and registering the kit. You'd provide it, it's done through a saliva sample, takes less than 10 minutes to self-administer. You can see the photograph, there's that little plastic tube got a plastic film with some liquid in, and so you put the saliva into the tube, and then when you close it up, the liquid falls into the tube as the sort of transporting liquid. But it's very straightforward in terms of saliva and filling it in. And then it's returned to Cynogenetics using the packaging with a prepaid label. The results are sent to both you as a participant and to your healthcare provider if applicable, and then genetic counseling is offered. And then you'll be contacted through after the consent to be contacted for ongoing or future research opportunities. And then how do I find out more or sign up to participate? The website's there, www.thinkpcd.com. And also the details of our phase 1 trial are on clinicaltrials.gov and accessible. And then there's some people to contact if you want to speak to a person, Candy Hirschberger or Stephanie Castillo with questions on that number. So I think that is all I have to present. I'll just ask Patrick if he wants to add anything, and then we'll move into Q&A.
No, nothing specific from my side right now. I think we got some great questions coming through the Q&A.
There are just a couple of things that I'd like to add to set the stage or help kind of explain a few things. First of all, apologies for these captions. We recently added an AI component to our Zoom and didn't know it would do that, but it's been very interesting what the AI thinks that we're saying. So we'll try to figure that out before we have another webinar. There's a couple of things that I think will be helpful for the community to understand, and they kind of run together. One is the notion of biallelic and what that means. So Dr. Matthews talked about alleles and what they are. And essentially, this is your version of a gene that you've inherited, one from mom and one from dad. And for most of PCD, that is autosomal recessive, we always think it's easiest to think of this as one is enough. If one of those that you've inherited works, you will not have the disease. It is produced as a recessive disease only when both alleles, the one from mom and the one from dad, are have pathogenic mutations and don't work. That's when you run into trouble. And the reason that this is important in this discussion is that we have a lot of people being told they have confirmed genetic diagnosis of PCD when they really don't have pathogenic mutations on both alleles in one gene. Excuse me. They may have one on one gene and one on another gene, and they're being told that's a confirmed diagnosis, and it isn't. So if you can think of this, it doesn't mean you don't have PCD. It just means that those genes don't confirm it. Your genetic test doesn't confirm it. So that may be one reason that somebody that already has a test that they've been told they have genetically confirmed PCD may want to still participate in this program and upload their test so that it can be reviewed and it can be confirmed. But if you can keep in the back of your mind that one is enough, if you have one working copy on any of these genes, then it shouldn't result in PCD. So I just wanted to say that for before we start the discussion.
Yeah, that's a great point. And if you may be in that category or just want to know, then part of our team's job is to review those reports as they get uploaded. And as John mentioned, there's genetic counseling resource. So sometimes the reports can be really challenging to read. We've tried to design ours so they're really clear, but if your report seems like it's in another language and you'd like some help just confirming your understanding of it, that's a resource for that as well.
Okay, I'll try to get through the questions here. Why are you targeting DNA I1 with only 10% prevalence and not DNA H5 with 26%?
That's a great question. I think what happened here was when ReCode formed, it really came out of the merger of two scientific efforts. One was to have the discovery about the lipid nanoparticles, the technology that allows the delivery of what we call payloads, which can be a messenger RNA or DNA into the body, but into the right cells in the body. And so Dan Siegwert's lab made the discovery and had this tissue-specific targeting with the lipid nanoparticle. There was another company called Transcriptics, which had the mRNA technology. I think DNA-1 is a smaller protein, and I think at the time, they were talking 8-10 years ago, the idea was that perhaps would be the first one, the better one to start with. There were also some models, some DNA-1 knockout models that allowed some of the preclinical science to advance to help that program go forward. So I think DNA-1 gives us a really good chance of what we call proof of activity, proof of concept, to really show that this method of bringing the instruction into the airway cells into the lung and have something that can restore function that's clinically meaningful, we have the best chance of doing that with DNA-1. And then as from that proof of principle, we'll then be able to expand through and to cover as many of the additional, because in principle, there's no reason why DNA-H5 can't be done in the same way. But generally, pharmaceutical investment comes once de-risking has happened. People like to see that when they're going to put their effort in, it's all going to be successful at the back end. So I think the PCD community are now at the point of which we can really show that we can do this, and then that'll bring forward more investment and the ability to go through. And frankly, ideally, we would treat the entirety of PCD. There's no reason why we couldn't do that. But I think we need to be realistic that in the first instance, clinical trials need to happen in a staged fashion, and the de-risking process is what's going to allow for further effort.
Yeah, I think we should think of this as the entry. This DNA-1 is the entry into this whole field of gene-based therapies, and we really want to have a very successful first time out. So that was the gene that seemed to be the most likely to be successful, and then we'll move on from there. And that's why we can all get behind it, even if it's not our gene. We have several questions related to will programs like this be available in other countries and for children under 18?
So our pediatric program will start as soon as we've got the potential benefit. So proof of activity in adults is generally what happens in drug development, such that we can then move down into younger ages. Pediatric plans have to be written as soon as you have adult data, and the earliest adult data, which we now have. So my job is now this year to start writing the pediatric plan. And as soon as we can start showing that we have some benefit, then the risk to applying a novel therapeutic into children becomes allowable. And so the aim is to be able to get this treatment down to the age of 1 or 2, such that we can prevent any lung damage happening right at the beginning. But for safety reasons, we all start in adults. And for the genetic testing plan, I think as soon as we've got that proof of activity, we would open up and we'd start to do younger ages. I think it's also worth noting that in the UK, they have included PCD in the newborn screening genetic panel. So I think as we get more resources behind this, I don't want to speak for the US, but I'm sure it would be a good idea if the US started doing newborn genetic screening, including PCD. So yes, pediatrics is firmly front of mind, but it will be a staged approach.
On the country, there's a couple questions about different countries and eligibility. One of the constraints that we always have with programs like this is to offer genetic testing in a particular country. There's a lot of regulations that we need to comply with. So the program is live in the US, and we know there's a lot of people who could make use of it in the US. For individuals listening who are in other countries, I would say we would be really happy if you wanted to reach out to us and just lodge your interests, because one of the things that we want to think about from a program perspective is how we best use the resources we have to expand it. And also, to John's point, some countries have very good existing genetic testing infrastructure, and we may be able to just signpost you in the UK, for example, to get testing that might already be available and help navigate whatever systems there already are before this program expands or doesn't expand to those countries. We just want to make sure that people get access to testing wherever they are. And that will also help us figure out if, for example, you've tried to access testing in the country that you live in and you've run into problems. The more we understand about what problems you're having, the more we can work from our perspectives to try to improve that, whether it's from bringing the testing program over or reaching out to the relevant government stakeholders to say, hey, this is a really important thing that that you can be thinking about. So I'm happy to provide my email in the chat or anything like that, and just say, please reach out, and I can put you in touch with the right people on our team to talk about that if you're comfortable.
Just so everyone knows, we have attempted to get on a newborn screening panel, not a genetics panel. And in the US, it is the National Institute for Child Health and I can't remember, NICHD. They make a recommendation, but it is up to each state health department to determine what's going to be on their newborn screening panels. So it is a huge undertaking here. You have to go to 50 different regulatory bodies to get a inclusion decision, and they mostly want to know that there's a treatment that will make a difference before they will include you in a newborn screening panel. This is, of course, ridiculous, and patients want to know what's going on with their children, whether there's a treatment or not. But that is that is still, from what I understand from a project we're doing with CZI, that is still the big limiting factor. You have to show that you have a treatment that will make a difference. We may have that now, but in the past we have not had that. So that has been a kind of a catch-22 for us. We have a question here about a research genetic test. So not a CLIA approved lab, but a test that was done during research through the GDMCC studies at UNC. Would those people be eligible for the trial?
Would you like to take that, John? Or
Yeah, if you could take that, Patrick.
Yeah, so generally speaking, the trial will require some clinical grade genetic testing. It may be that the testing that's been done is research grade, or it may be that it is clinical grade. I think the safest thing to do would be to upload it through the platform, and then our team will be able to make a determination of whether the sequencing is of the quality that to the flow diagram that was showed, you could go straight through to the trial without needing to be tested again. Or if it was a research grade test, then we typically recommend a confirmatory clinical grade test that would happen through the program.
We have a question about the slide 5 with the 65 to 80% of undiagnosed patients are anticipated to have an identifiable disease-causing variant. So there's no bias in diagnosis that we would expect along the lines of severity. More severe cases are likely to have an identifiable variant, while unidentified variants may account for less severe disease. I can give my opinion on that. I don't think so. I mean, I think we're not finding people not because their disease is severe or not disease, it's because nobody's looking. So a lot of these people with very severe disease in bronchiectasis clinics have not had genetic testing for PCD.
I mean, I think the question asking why isn't that 100%? And so I think the more we understand the clinical phenotypes and do more genetic testing, we'll find out where that uncertainty is coming from.
Yeah, and I would just add to that, one of my view is also the value of programs like these is before you have widespread access to testing, you get really biased data, and it's hard to make sense of it sometimes. Is it more? Is it less? And I think the truth is, the more data we have, the more we'll be able to answer those kinds of questions accurately, and broadening the group of people who can get access to testing is really important for this.
Okay, so is whole genome sequencing recommended if a PCD panel comes back negative? I'm actually going to see if Dr. Lee would respond to that with some of her experience with some of the whole genome, whole exome sequencing.
Well, I think that would be, if possible, would be a next step, but I would not go into that without a geneticist or a very good genetic counselor working with you. This is what we do on a research basis when we're trying to find new genes. But on the clinical spectrum, I think you just got to make sure you've got somebody who knows how to interpret it.
That kind of plays into the second part of this question. Should patients with a clinical diagnosis of PCD, where there's not genetic confirmation, be concerned about not having genetic confirmation at this point? I'm going to just jump in and say yes.
Yeah, you jump in and then maybe Dr. Lee.
I don't think you should be concerned, but I do think the way things are going and kind of the soapbox that we've been on through the PCD Foundation for, a decade plus, is that knowing your genes is not just for diagnosis. It's going to be necessary for precision medicine, and that is the way medicine is going. So there's not a downside to trying to get genetic confirmation. It may not be possible because we don't know all the genes and variants yet, but it's definitely worth trying to do. But I feel like I should let clinicians weigh in here too. That's just my opinion.
Well, more information is better, I think. But the problem is some people think that a negative genetic result makes their diagnosis in question, and it does not. A negative genetic result just means that at their present knowledge level about genetics, we cannot confirm in that particular patient. But as we learn more about different genes and different variants that are mutations, then we will be in a better position to confirm. So there are people that over the years that we've been studying in the GDMCC, that our initial genetic tests were what we called uninformative, meaning that we couldn't say that they had PCD based on those tests. But then later, when other genes were identified, we were able to confirm. So it's not going to say you don't have PCD. It's just going to say we can't at this point tell you what your genetic basis is for PCD.
Yeah, I think PCD is not an undiagnosing program. That's not the point, and it's not even possible to do that. So, and that's true with all genetic testing. So there are other ways to make the diagnosis. Genetic testing is just going to be important for some of these personalized medicine options. Okay, we have a question here from Alice. Is there anything known about if there are patients who have low NO DNA-I1 mRNA protein expression, but is being caused by genetic mutations in another gene, a promoter of DNA-I1, for example? So would these patients benefit from these trials? Is there a way to detect and include them?
I'm happy to take this one. My PhD was in non-coding DNA and its role in rare disease, and so I thought a lot about these kinds of questions of promoters and enhancers and introns and other things that don't affect the protein itself, but may affect where it's switched on and off. And John did a really great intro to this, that these are typically mutations or variants that result in loss of function of the protein. So there's no protein or not enough protein being created. And so this could happen with a mutation in a promoter, for example, or an intronic splice site. So these are things that we're really careful to look for with the exome sequencing that we do. If you're familiar with the technology, though, you'll know that it isn't whole genome. It focuses on the coding regions, but it also includes targets that go for promoters and other non-coding regions. So it is possible that there are some very distant non-coding variants that we're not aware of and wouldn't be described in any of the known population databases that wouldn't get captured. This is a fundamental limitation in most genetic studies these days. One thing we have done, though, is there is consent in the program for if you'd allow for your sample to be stored, to be reanalyzed at a later date for research purposes with something like a whole genome. And so there's a potential future opportunity here where we can look at individuals who don't receive any causal or contributing genetic variant here and see if there are some of these kind of even rarer variants that are contributing. But this isn't something that we're necessarily able to do in the near term, but it is something that we thought about from the design of the program as a future direction that it could take to make sure that these are captured if it's at all possible.
We have a question about when will people with DNA age 5 know that their gene is up when we're ready to do a study with them?
I think we need to see this year that the DNA1 results come through in the clinical development program. In the background, lots of diligent preclinical work is happening. So things are being moved along. The aim of ThinkPCD is to have, as soon as that trigger is pulled, then all the people that can be notified, because that's relevant for their genetics, will get to know as soon as possible. So I can't promise a timeline, but I hope that it is in the relatively near term, versus sometimes when we're talking about research and people talk about things happening in 5-10 year horizons. I think we're much closer than that. I'm an optimist, but I think if we can get through these clinical programs in the near term, it will trigger a lot more clinical trial work.
And certainly we will be telling people about opportunities that come up, and you can always check clinicaltrials.gov where all current research on PCD is listed, and you can see if there's anything there that would be of interest. We have a question about a follow-up on the promoter intronic region question. Are there any identified epigenetic causes for PCD?
Yeah, I'm happy to take this one. As far as I'm aware, there's very little, and I'm taking this question as asking about major methylation changes, imprinting, those kinds of things where some genetic diseases, this is a hallmark feature. As far as I'm aware, it's not a major factor in PCD, but it's not something I'm an expert in. So I will definitely do a little work after this and make sure I'm correct on that. But based on a quick literature search while I was seeing that question come in and my knowledge on this today, I don't think it's a major factor here.
Dr. Lee, would you like to weigh in on that, having spent so much time looking at PCD genetics?
I don't know of any epigenetics, but I defer to Mamuna, who I don't think can speak at this time. I don't know of any epigenetic causes.
Okay, well, I think that brings us to about the end of our questions, unless there's anything else from anyone. If not, big thank you to our presenters today. We really appreciate you taking the time to share this information with the community. And we will send out a follow-up. This will be posted, this webinar, as soon as we can do it. It has to go through a little bit of work first, but we'll post this, and then we will also send out notifications with contact information and places that you can look up more information about ThinkPCD, about other clinical trials, and about cytogenetics. Thank you all very much.
Thanks everyone. Appreciate it.